to Lunch with Docs HD with Dr. Claudia Testa today. She is from VCU, that's Virginia Commonwealth University, and she is an MD, PhD, professor of Department of Neurology and Parkinson's and Movement Disorders Center, also Huntington's Disease Program Director, and Joan Massey Chair in Clinical Parkinson's Disease. And we are so fortunate to have her today. She's actually on vacation and she's devoted this time to us. So we're so pleased to have you today, Dr. Testa. Our topic today is going to be clinical trials for Huntington's disease. And I also would like to thank PMD Alliance for providing this platform for us and Teva Pharmaceuticals for funding. We appreciate your time and resources to help us bring information to our HD community. So welcome, Claudia. Thank you for joining us today, and thank you for taking this time out of your vacation to spend with us.
What I want to do for the slide portion is to talk about recently wrapped and ongoing current clinical trials and use those to also introduce that are really important for people to understand as new clinical trials come out. Then it gives you a framework to think about whatever new is coming down the pipeline, in addition to understanding the results that are coming out now. So next slide, please. Perfect. So this is my one big disclosure. I've been part of the Huntington Study Group, which is a nonprofit academic network dedicated to seeking treatments that make a difference for Huntington disease. Through Huntington Study Group, I've participated at many levels in lots of different clinical trials. I'm currently getting paid through Huntington Study Group as a site principal investigator for the Signal study. And I actually am also currently getting paid through Huntington Study Group as a Huntington Study Group consultant to Unicure regarding their protocol design for upcoming clinical trials, and we'll also mention those. I do not have any other financial or potential conflict of interest disclosures. All right, next slide, please. So one of the core things about thinking about clinical trials in general and new therapeutics is what is the goal of the new therapeutic? We all want a cure for Huntington disease, but that what a cure looks like may be different patient to patient. And one could achieve something that's justifiably within the realm of cure by targeting much different things. So some of the ongoing clinical trials are targeting specific symptoms, whether they're motor or non-motor. They're looking at an overall outcome. What good is it if you make a symptom go away if your overall functional status or your overall quality of life doesn't change? So looking at big picture outcomes, whether or not something actually protects brain cells, or if it is what we call symptomatic care, the things in the brain keeps changing even though the symptom control is better. Are you changing the rate of how symptoms progress, whether that's internal or external? And the big prize is changing, even improving what's happening within the brain as Huntington's disease goes on across the lifespan. Next slide, please. So when we talk about outcomes, I mentioned functional outcomes. Clinical trials are designed to try to figure out if a new compound is safe, first and foremost, and then to figure out if a new compound is effective, actually does something. So you figure out if things are effective by using outcomes, and you generally pick the outcome ahead of time. So you have to say, this is the outcome we think, and this is how we're going to test it. If you don't know what your outcome is ahead of time, then you might not have enough people, or you might not have the right kind of people or symptoms in order to adequately test something. So people pick their outcomes at the beginning of the trial and design around that. There's an interesting shift going on. This is a very nice review from 2014, looking at all the published clinical trials using it's called pharmacologic, so medications pills or infusions, something like that, beyond exercise or non-medication things. Most of them use a clinician-reported outcome. That means the neurologist or the psychologist or whoever gets to some used a patient-reported one, and very few used an observer, like a caregiver, family member, friend, someone like that. However, there are lots of Huntington's disease-centered measures or observer-reported under development, and we're starting to see a shift in what people are looking for, whether community or people means the FDA. We're starting to see a shift in what people are looking for as like a mix of outcomes reporting from lots of different perspectives. So I wanted to start with this concept because it'll come up pretty quickly in reviewing recent clinical trials and what the results were. Next slide, please. So Huntington's disease has a big range of clinical features, a lot of them non-motor, which we'll get to. I want to start with motor signs and symptoms. A classic motor sign that impacts a majority, although certainly not all, of people with Huntington's disease is involuntary movements. The most classic is chorea, which is from the Greek word to dance. Dystonia, another movement that can be either very active or very fixed, is another classic involuntary movement. But there's quite a big range of motor signs and symptoms. And these can have highly variable impacts on people's day-to-day functioning. So for some people, for example, wiggling a little bit, it doesn't really matter to them. They get through their day and they do just fine, and they can do everything that they want to do, and they don't really care if other people see them wiggling, so that doesn't bug them. Whereas other people find that their amount of extra movements makes it much harder to do basic tasks or to walk around, do their hobby or work. As another example, being able to move your eyes around quickly from object to object, to pursue moving objects, or to jump from one to one, which is saccades, is really important when you're driving, to be able to move your eyeballs around, to look at the other cars or the other things in your field of view. Motor impersistence means it's more difficult to hold posture or to hold emotion. So we often look in clinic at people sticking their tongue out and seeing if they can do that for a really long chunk of time. You can think about if you're in the car and you're at a stop, you want to keep your foot on the brake in the same place for an extended period of time and not have the car kind of go like this because you're moving back and forth. So some of these are things that are really easy for both the patient or the family members to pick up. Others are things that people don't come in and tell me about. They don't say, my oculomotor control is really annoying me today, but may have an impact on function. So let's review motor signs and symptoms and how people are trying to think about them in clinical trials. So let's go through some clinical trials really focused on motor features. Next slide, please. Oh, I forgot this slide was here. So this is what I'm trying to convey when I talk about motor signs and symptoms and how they can fold together into impacting function, whether that's driving, communicating with other people, walking in balance, or the bigger picture things like what kind of job you're doing, if you're working full-time or part-time, what kind of responsibilities you have at home, and if you're getting to all the activities that you want to do. So many of the most recent trials is what's called a primary outcome, say a very specific motor sign like chorea or involuntary movement, and then a secondary outcome, they look at their primary outcome first and use that to decide if the new agent is working, and they look at the secondary outcome next, like an overall functional target or an overall quality of life target, to see if a change in something like chorea or a very specific sign or symptom actually translates into a useful gain in overall function. Next slide, please. So these trials wrapped relatively recently, First HD and ARC-HD. This is part of my Huntington study group disclosure. I was the paid global co-principal investigator for these studies. They're quite close to my heart. They were looking at a compound called deutetrabenazine, which is the tetrabenazine molecule reconfigured to have deuterium, which are the blue Ds on it, that makes a stronger bond between the carbon molecule and the deuterium molecule compared to the hydrogen molecule that's usually there. That slows down the breakdown of the active metabolites. So in theory, you get a longer action and lower peaks, so less peak dose side effects was the concept. This was a primary outcome of chorea, or involuntary movement. So improvement in chorea was the key thing. But then there were several secondary outcomes. Next slide, please. So the deutetrabenazine, the brand name is Austedo, is one of the several things now in this growing class. So there's the regular tetrabenazine, which is a generic, and then xenazine is the brand. The deutetrabenazine, which we're talking about now, which is Austedo. There's also now valbenazine, for example. So there's 4 things in this class now. The idea was of all of them is to reduce involuntary movements or chorea. And that's what the FDA approval was based on. Do we succeed? Yes, in reducing chorea. I wanted to point out that this was a key secondary outcome, patient global impression of change. So we used a patient global impression of change and a clinician versus global impression of change. And we used the patient global impression of change as the upfront key secondary endpoint. So after we looked at chorea, the first thing we looked at was patient's impression. Somebody wave at me if you can't hear me anymore because I'm getting little nasty messages from my internet. The bars that say PGIC under them, this is where we ask patients, compared to before you started the study drug or placebo, how do you feel like your Huntington's disease symptoms are doing? Are they worse? Are they better? Are they the same? The blue bar is the percentage of people who said they were much or very much improved compared to the beginning of the study. And they were taking deutetrabenazine, or SD809. The orange bar is the people on placebo. So you see a lot more people on study drug, on deutetrabenazine, thought they were much or very much improved compared to the number of people who thought that who were taking placebo. This was actually a bigger split than we got when we asked the clinicians for their global impression of change. We asked the neurologists and the psychiatrists who were at the sites, what do you think about the person's overall Huntington's disease? There was a clear difference between the deutetrabenazine group and the placebo group, but it wasn't as big a difference as asking the patients themselves. So I think this was quite nice because there's a lot of controversy over whether Huntington's disease patients themselves really recognize symptoms consistently and can give a consistent and clear take on their own overall state. So I see this as a very nice, empowering outcome. And I know other studies are looking at adding or including these kind of global impressions or patient or caregiver reported outcomes to supplement clinician reported ones or even trump them in some cases. Next slide, please. So it's terrific that we now have multiple things, deutetrabenazine, tetrabenazine, and others all in that group. An area of study that's only really getting underway in Huntington's disease research and clinical trials is relative benefit of these new things to older and much cheaper stuff. And how do people get access to these? Is the access barrier worth it? I think that this question will come more and more into play as hopefully we get more and more cool things that hopefully are not inanely expensive for people to access. So the Huntington Study Group just awarded 3 C funds. One of them was for about talking to kids about Huntington's disease, but 2 of them were about the relative benefit of deutetrabenazine, tetrabenazine, and others against the older compounds that were not specifically designed for chorea or involuntary movement control in Huntington's disease. So to highlight that our work isn't done once we get something approved from the FDA for a treatment to make a difference for the Huntington's disease community. It has to not only be safe and effective, but be reasonable to access and feasible to access for all the people who need it. And it has to have enough of a gain over what used to be out there for it to really be a great choice for lots of people. Next slide, please. So recent clinical trial that was closed was the Amaryllis study. This was Pfizer and the Huntington study group. I forgot to put that on here. This was reported out as a negative outcome in late 2016. They were looking at motor symptoms generally. So instead of just involuntary movements, they were targeting a wide range of motor symptoms. They used other exploratory, they called them, measures just to see what might be happening if they could get an improvement beyond motor. They decided to close down the open label arm of the study and not continue to pursue this agent. So why am I talking about a trial that we use the word negative for or that got closed? Are there any upsides? Well, this is a very, very nice study design, which included a lot of biomarker work. We'll talk about biomarkers in a minute, and secondary outcome work. So lots of valuable information on how Huntington's disease works, the role of these phosphodiesterase inhibitors in Huntington's disease, and what people might include in the future for similar kinds of studies. So accelerating the design of the next realm of clinical trials. Next slide, please. Another trial with a negative primary outcome result in motor signs and symptoms was PRIDE-HD, which was looking at pridopidine. This showed no clear gain on the total motor score. So again, looking broadly at motor signs and symptoms. This closed down at the very end of 2017. They still don't have all the published data out yet because it's closed relatively recently. So again, why talk about something that has the word negative in here? Pridopidine did appear to slow down functional change over time, and the main upside for this one is there was a very high ratio of people with active drug versus placebo. So a lot of times people say, I don't want to join this study because lots of people are going to get placebo. I don't want placebo, I want the stuff that works. The thing with clinical trials is that we don't know if it works or not. There's yet another example of something that didn't actually work, but needed to be tested for us to find out. One of the reasons that this was a negative outcome result was that there was a huge placebo effect, which is a little unexpected, but everyone in the trial, all the site investigators and all the participants knew that 4 out of 5 participants were getting active drug, and only 1 in 5 was getting placebo. So the theory is that everybody just said, I must be on active drug. It's a 4 in 5 chance. And so the placebo effect that's normal got really amplified. So now we're trying to convince people it's not a present to the community to have really big treatment groups and really big placebo groups. In fact, we should stop calling them treatment groups because we don't know if it's going to work or not. We should just call them active drug groups versus placebo groups, because the goal of the clinical trial is to figure out if something's working or not as fast as possible. So if it doesn't work, we can move on, and if it does work, we can get it out there. So there's now a worry that the pendulum is swinging back to having more even active drug and placebo groups, in part because of what happened with the data in this trial. So that's been quite valuable to clinical trials generally. Slide, please. So those were all clinical trials that completed and went all the way through FDA approval or completed and closed out for negative data very recently, and they were all focused on motor signs and symptoms. We all know that there's a huge range of other clinical features to Huntington's disease, and that cognitive and behavioral symptoms can often be the biggest ones for patients in terms of impact on overall quality of life and on overall function, whether that's shifting what people are doing day to day in ways they don't like, or impacting things at home or work, changes in mood, changes in behavior, and changes in cognition are a huge thing that clinical trials are finally turning to in terms of looking for targets. Next slide, please. So this is an ongoing study that I believe just ended enrollment. The closest site to us is University of Virginia, and we just sent them someone a couple months ago. So I'm pretty sure that the study in general is just closed. This is an ongoing study for a novel agent addressing irritability, that short fuseness, the snappiness that then like bang resolves just when the person you're snapping at is really ready to have an argument. That's a really corrosive and also really common symptom in Huntington's disease. So I'm quite pleased to see the National Institutes of Health and the NeuroNEXT network take on something of big importance. Study comes out. Oh, I'm getting the little high sign again, so I'm going to put my earbud in and see if I can do this more smoothly. Oh, no, it's gone away again. It's like a trick. Okay, next slide, please. So we've been talking quite a lot about clinical about so far have exclusively worked with patients who have clear clinical diagnosed manifest Huntington disease. Some of the stuff I'm about to talk about is testing experimental agents or active drugs in people. So many of you, if you've participated in a research study or a clinic visit at Huntington's Disease Center, have gone through a motor symptom sign exam. You tap your fingers, you look around, stick your tongue out, you walk, do all this stuff. In clinical trials, there's this thing called a diagnosis confidence level. So I use a lot of fancy language to basically say, how confident are you that the exam, that the stuff you're seeing, the signs you're seeing in the person represent Huntington disease and not something else? So as a basic example, you may score the person as having a pretty bad gait, but they may have a broken hip, right, which is not Huntington's disease. So it asks you to say, this exam was perfect, normal, 0, less than 50%, getting confident, getting in a 3, or a 5. This is clinical Huntington's disease, no doubt about it. Many of the ongoing, I was talking about prodromal and what that means. And the reason I'm belaboring it a little bit is because this is a really key thing for the current enrolling clinical trials. So prodromal means you have a little bit of signs and symptoms, but not enough for a diagnosis. It means you have a little bit of signs and symptoms, not 0. A lot of the people who are coming in to say, hey, I hear this thing is enrolling, I really want to participate, I want to do my thing, have no signs or symptoms. That's non-manifest, no signs or symptoms. Prodromal is you do have a little bit of signs and symptoms. Just really not enough to call it a diagnosis. You might trek along there for quite some time. So that's what people are looking at. Early usually means that you have a clear diagnosis, but you have a small amount of signs and symptoms, and functionally you're doing pretty well. Working, running the household, all that kind of stuff. Next slide, please. So if you have people who have very, very little going on in terms of symptoms, just a little bit of this or that, prodromal people, or even people who have a clear Huntington's disease diagnosis but have very early symptoms and mild symptoms, how are you going to tell if anything changes over time? By definition, these people are doing functionally pretty good. So you can't, do, maybe you can measure a change from functionally pretty good to awesome, but, I'm not sure how you would spec that out. So one of the things people use are biomarkers. So this is a fancy word for things that can actually be quite simple. Anything, whether it's brain images, compounds like proteins, or just a pen and paper test, but something that correlates with, that tracks with or predicts something that patients care about. Disease progression, symptom severity, for example, and changes on the brain images, and show differences in that use that as a biomarker to predict that this is doing something positive for patients, even though there's not much to see on the outside. Next slide, please. So I'm going to talk about a couple of things that are a little bit old school, and I'm going to be brief on them because I know people want to talk more about the new stuff, but just to show how people are working with manifest or prodromal or early things. So the two studies that did this first were you're looking at coenzyme Q10 and at creatine. Next slide, please. So with coenzyme Q10, it tested the safety of coenzyme Q10 in people with a mutation, people actually participating in the PREDICT-HD observational study, who had no symptoms, who were non-manifest. This was huge to get this approved because you're basically doing something to people that could have some risk, but they have no symptoms. So just getting this to happen is the foundation that a lot of the current studies rest on. So that's a purely safety trial. PREQUEL, the trial with creatine, was looking at whether creatine treatment compared to placebo slows down biomarkers, in this case imaging, in non-manifest subjects with a mutation and also people at risk. Next slide, please. So coenzyme Q10 was safe at all the doses it was tested in, so that was great. Creatine also went through a huge phase 3 trial in people with symptoms in Huntington's disease that was a negative outcome, so we do not recommend creatine to slow down or change disease progression in people with symptoms. What's quite interesting is in the PREQUEL trial, they looked at people who had mutations and also people at risk who didn't have mutations, and they showed a slowing of atrophy, brain change on imaging in parts of the brain in the people who took the creatine compared to the placebo. This was not benign. A bunch of people discontinued because they had terrible GI side effects and really hated all that creatine, and only 2 of those people were on placebo. The other 13 were taking creatine. So it's not benign necessarily to ask people with no symptoms or with even no mutation to do something, but this is also, despite the fact that it's super small and it doesn't prove that creatine works because it's too small to do that, it's being pointed to by a lot of the current studies in order to get regulatory approval to go ahead and have participants who are prodromal or even non-manifest actually take on experimental therapeutics and take on risk. Next slide, please. So right now, there's something that people who are prodromal, so that diagnosis confidence level of 2 or 3, a little bit of something, but not enough for a diagnosis, or very early Huntington's disease, a clear diagnosis, but doing great functionally. I'm usually personally operating at roughly a total functional capacity of 12 myself. So this is really implies that people are out there working, running their households, doing their thing, whether or not they have a Huntington's disease diagnosis. This is looking at monoclonal antibodies, so it's a monthly infusion compared to placebo. And they're looking at overall function, lots of different symptomatic and sign endpoints in motor and cognitive. They're also using lots of imaging measures to hopefully drive this with biomarkers too. They're hoping that this is a neuroprotective, so protecting brain cells type of therapeutic. Next slide, please. So here's a bias alert. I am part of Virginia Commonwealth University, and we are a site for Signal, and we are actively enrolling. The study will pay for people's transportation or even hotels if that's necessary. Clinical trial participation should always be free to the participants. Heather Ward, that's her work cell and her email. If you're not in our region, great. We'll happily help point you to another site. You can go look yourself on the Huntington Study Group website. I put up the web link that Melissa can get out to everybody as well. or if it's confusing or you have trouble reaching people for any reason, you can just reach us and we'll hook you right up with whatever works for you. This is enrolling a second set of people because the first round went so well that they're really pushing to get a bigger second cohort in because they're excited, and we're hoping that means that this compound is going forward. It's still an experiment. We don't know the answer yet, but their first round got them some really hopeful information. So I think that that is super exciting. Next slide, please. So this study is ongoing now, Legato. It's no longer enrolling. Enrollment has completed. So that's actually also super exciting because it means that we're that much closer to getting results out of it. So this, again, is looking at lots of potential endpoints and has lots of science built in. This is another Huntington study group, European Huntington Disease Network collaboration, and the sponsor for this one is Teva. Next slide, please. So I'd like you to please focus on the bottom of the slide. These are disease-modifying agents. So we think they're going to protect neurons or change the course of disease overall. The one at the very bottom, the VAX-NEXT study, is the signal study. In yellow, it's highlighted. The second cohort is enrolling right now. The Quinimod or Legato one above it is the one I just talked about. The other ones that are neuroprotective ones are tiny little ones, mainly in France, that are not available for enrollment. And then at the very top, the irritability study that's ongoing is the one I mentioned earlier. It's the symptomatic one. There's also one, I think, in Australia that just has one site looking at smoking cessation. So these are things that are that I've gleaned off of clinicaltrials.gov, HD Insights through the Huntington Study Group, and some of the company websites as what's going on right now in terms of symptomatic, neuroprotective, and disease modifying. And it's just that last one on the bottom, signal, that's enrolling right now. Next slide, please. So I want to very briefly mention the land of stem cells because people ask me about it all the time. There is one kind of in prep, slightly ongoing thing using stem cells in the US called HD Cell. This is at UC Davis. I believe Vicki Wieluck is the principal investigator. This is using stem cells as like payload delivery systems, like little factories that go in. So these crazy people are using brain surgery, stem cells, and growth factors all at the same time. So the idea is that they have their stem cells trained to make lots of growth factor, and then they inject the stem cells directly into the brain in order to have them get in there and do this. This study is being pointed to by some of the studies we're about to talk about because it's actually using brain surgery in Huntington's disease to deliver something experimental, and other groups are going to do that too. And they're trying to think about how to move as quickly as possible. So they're trying to observe people way before the surgery and use those outcomes to make a faster turnaround for their trial. Next slide, please. So right now, there's people in the initial observation study called PREQUEL, and then they're going to use that to anchor how they're going to do things on the other side of the surgery. This is a design that's being really closely followed as people make new things. Next slide, please. So again, very briefly, I think we talk a lot about experimental therapeutics, stem cells, genes, things that are really high tech. And sometimes we miss opportunities that are lower tech. So I think there's still a huge amount of opportunity in exercise or movement, non-medication therapeutics. This is one of my favorite papers of all time, Dance Dance Revolution in Huntington's disease. As an example of, there are creative ways to impact people that don't involve pills or infusions. Next slide, please. And I think one of the areas that we're really missing out on that other neurodegenerative disorders are already really into is more cognitively based non-medication interventions and studying those to figure out which ones actually do something positive in Huntington's disease. So here's an example of mindfulness training in Parkinson's disease, and they showed actual positive brain changes on MRI. So this isn't just like new agey, you feel better because you've done your mindfulness. You can actually impact brain cells with this stuff. So I really see some of this as what we're going to see people invest in the next 2 to 5 years, looking for things that you can do immediately and proving how they make an impact. Next slide, please. In the meantime, what everyone is investing in the next year is the high-tech stuff. So this is a picture from well over 25 years ago, probably 30 years ago, in Venezuela, in a little tiny town in the Maracaibo region. There's a kid and her mom, and in the background, the sister, the aunt of the kid, coming in from their annual research visit. So this is research participation. Kid's got her best dress on, as her annual picture. Behind her is the late Jack Penny. He and many others, Dr. Nancy Wexler, Dr. Ann Young, founded the Venezuelan Research Group. That's next to handing him something is his daughter, Ellen, who is now an MD, PhD Huntington's disease clinician and researcher, at least many years later. So one of the key things about Huntington's disease that really draws all of us in is that this is a family affair. And we are finally seeing the experimental therapeutics go in that direction too. Next slide. So Huntington lowering, so lowering the amount of mutant Huntington protein is the really hot high-tech area right now, and where we're going to see the most action in the next year. So gene silencing is one way to do this. So everything starts with the gene and the DNA, where there's a mutation. The DNA makes a messenger RNA. The messenger RNA makes the protein. I've ripped this off totally without attribution from HD Buzz, in case anyone's wondering where this picture came from. And gene silencing is just one place in this pathway. You could imagine getting rid of the protein once it's there. You could imagine blocking that arrow from the DNA to the messenger RNA. There's one type of thing called gene silencing that blocks the message going to the protein. But everything in this pathway is really up for grabs right now. Next slide, please. So this is all of the things that I found that seem to actually be happening. You know, there's lots of things that you dig into them and they're not actually going on. So I broke these Huntington lowering agents into 2 categories. The top category is things called pre-mRNA. So the messenger RNA, so the DNA where the mutation is, that makes the messenger RNA. So this is kind of shutting down at the messenger RNA level, roughly speaking. These targets are antisense oligonucleotides, or ASOs, and the way they're delivered currently is several repeated injections into spinal fluid. Right now, most of the structures are monthly injections forever, but it's really unknown how often or how long injections would go on. The most famous of these ASOs is the Ionis one, the phase 1/2 wrapped, and it had great data, no safety concerns, which is awesome. The lower mutant Huntington protein in the spinal fluid, we don't know what that means. So it's cool that that happened, although we don't know how that translates into anything. So there is currently no word. I was just at an Enroll-HD meeting. There is, yes, the next trial is going forward. No, we don't know anything about the time or the sites. So that's the answer to pretty much every question I get about this study. Awesome data, great safety, don't know what lower mutant Huntington means. Yes, next trial, definitely happening. Don't know anything about the timing or the sites. Even the people who work for the company didn't know anything about the timing or the sites, but they're super excited to keep that going. One thing that is, looks like from clinicaltrials.gov it is recruiting, although I think it's somewhat by invitation only, is a little unclear on the websites, is the WAVE ASO. So this is similar to the Ionis compound, and it's also spinal fluid injections. It's ongoing right now. So exciting stuff that there's 2 different variants of things this going on. The main difference is that the Ionis Roche one, the one that just got through safety, it tamps down normal Huntington protein and mutant Huntington protein. And we don't know what happens when you tamp down normal Huntington protein. We think you pretty much shouldn't go to 0, that'd be a bad idea because it's doing something normal, but we don't really understand how far down it should go. So it's doing what it's supposed to do, a great thing, less mutant Huntington protein, but also less normal. And it's probably going to end up okay, as long as we don't go to 0. Don't know yet. The WAVE antisense oligonucleotide, the WAVE ASO, targets just the mutant Huntington. So their goal is to drive down the mutant Huntington, and they're going to leave the normal Huntington alone. So that's an interesting advantage. Their disadvantage is that they need people with very specific genetic backgrounds for their compound to work, to actually be specific like that. So the Ionis one has the advantage is that it can be used for anybody, but it's going to switch down normal. You're not going to be able to take mutant to 0. The WAVE has the advantage that they can try to take mutant to 0 because they're not touching the normal protein, so that's awesome. But it's not going to be for everybody. You're going to have a particular genetic background. So a little bit of very similar approach, but with much different advantages and disadvantages between the 2. So I think it's awesome that there's 2 companies that are very far in development with this kind of compound. Everything under the next bar, the 3 left, are things that haven't been put into humans yet. That's why it says preclinical, but they all have very public statements about when and how they're going to try to do that. So I think that those are actually moving. There's an XXX with the last one because there's no public name for the compound, so I don't know what to call it. The bias alert, I am working through Huntington Study Group with Unicure. All of these are going to be single injections, so that's a big advantage. You don't have to do something one.
What can I do now? So she wants us to stay healthy, pamper your brain, and be engaged. Enroll HD for the whole family. This is a clinical study that is for the whole family, whether you have HD or not. And the healthy individuals are the control for this study. And she says, bias alert. Dr. Claudia Testa is with VCU Health, so that is the site that she is at. And so please call Heather Ward for more information about clinical trials at VCU. And her phone number is 804-382-0076. You can also email her at heather.ward@vcuhealth.org. And also find the site www.enroll-hd.org/participate. Everybody should be participating in the Enroll HD study, whether you have HD or not. So just want to thank Dr. Claudia Testa again for this fantastic presentation. And thank you, PMD Alliance and Teva Pharmaceuticals. My name again is Melissa with the HD Support and Care Network. Every 3rd Tuesday of the month, Lunch with Docs HD. Everyone have a great month, and we'll see you on Zoom